Hi there, Ms. Lams here. In this video, we are going to highlight a few points regarding to the criteria A, focus and method in the IB Biology EE. So let's get started. So this is just a screenshot of the score band, and I have highlighted some of the key points in here. Later on in the next few slides, we'll show like summary of what these score bands have mentioned about. So you can pause this video and have a take a good look at this part. And then moving on to the next part. So this one is another section coming from IBO, examiner guidance, is that what they are looking for. So unpack the criteria. So I've again highlighted a few key points here. You can pause the video and read this through. Next few slides, we will talk about the summary of these two slides. Okay, so let's move on. So first of all, in section A, you need to look into the topics, whether the topics is suitable for your biology EE. So first of all, you will notice that this must be biology related and it must be focused. So for example, some of the examples of the topics you can see here, some of the focus topic and broad topic, because in EE, you only have about 4,000 words to cover the whole passage. So it's important to narrow down the scope of your study. So for example, some not good example compared to a good example, you can see that, for example, they were looking at, how does the detergent affect the environment? But this is very broad as to what kind of detergent you're looking at and what aspect of the environment that is affected by the detergent you're looking at. So you need to narrow it down. Okay, so you need to narrow it down. So for example, talk about detergent toxicity in soil bacteria. Okay, so they just focus on the soil bacteria in terms of the environment. So you need to have a more focus and narrow down your topics. Okay, so then you can cover the scope of your study within 4,000 words. And then next part is suitability. Okay, so a few things of IB guidance is that your experiment should not cause harm or death, okay, to the organism that you're testing. So for example, let's say you're looking at caffeine at mealworm. So before you conduct your experiment, then you should look at the safety concentration of the caffeine for the mealworm, so you don't kill the mealworm during the experiment. Okay, and then another key part relate to the safety. Okay, so for example, when you conduct your experiment, you do not breed harmful microbes. So how do we ensure that it's, first of all, you have a known source of microbes? Okay, so for example, let's say it's a probiotics, okay, pills. Okay, so we know that that's not harmful. Comparing to something that is like obtained from nature, so you cannot just do like say swipe your palm and then use those microbes for your experiment. So those are not acceptable. And then another one is do not use bodily fluid, okay, as some disease can be transmitted through bodily fluid. So some student may be thinking of how the saliva amylase in the saliva performance, okay, looking into something to that. But then you cannot use that because the source of amylase is from a bodily fluid, the saliva. So a few things to consider when you draft up your topics. Okay, so let's look at the examiner feedback related to this part. So if you look at the score band, you'll notice that it's separated into three parts: this topic, suitability of the topic, and then your research questions. Okay, so you should be very specific related to your research question, and then relate to the methodology. Now the methodology is not just list out the steps of your experiment. The methodology here is related to two parts. One is your research, how you do your research. We'll cover that later on. And the rationale, okay, so the explanation of how your methodology helps you to answer the research question or the topics. Okay, so and then some later on, as we go more other criteria, you notice that sometimes the strand is not equally weighted. But in this case, for section A, okay, for criteria A, the three strands are weighted equally. So the topics, the research, and methodology. So related to the methodology, you need to justify, as we mentioned just now, you need to justify the steps. So by justifying the steps, you will explain how this methodology helps you to answering the research questions. And then one of the highlights from the examiner feedback is candidate need to look more closely at the positive and the negative control. Later on, we'll have some videos to show you what we mean by positive control and negative control and why they are important. This will be very useful when you use to construct a line of argument. So to support how your methodology helps to answer questions, to have a valid experiment. Okay, and the next one is the second part is how in this referring to the method part as well, is how you select your published resources. Okay, so your references. And so first of all, you need to show how you obtain those from a reliable resources. And then later on in criteria C, then you need to assess how useful these resources are. So we'll come to that later on in the next few videos on criteria C. Moving on. So this is a PDF. Okay, you can see a PDF in the video description link below for the full PDF. So this annotation, PDF annotation, is done by my colleague, Miss Rawat. So let's go through a few things of that. Okay, so if you download the software, the PDF, you will notice that, okay, so there is actually some EE guidance. So they will talk about the choice of topics, okay, must be biology related. Some of the inappropriate topic we've just covered, like how can we prevent the harmful bacteria from breeding, okay. And then if you do it human subjects, if your experiment is related to human subject, then you need to have a consent form, okay. And if your test subject is underage, that means under the age of 16, then you need to make sure 18, I think, under the age of 18, then you need the parent's guardian to sign those consent form. So yeah, we'll also mention about bodily fluid as well. Example topics, okay. All right, treatment of the topics. Preliminary research is based on your experimental data, okay. Survey is also regarded as some of the primary research. And then secondary research is from the publications that you use, okay. Supervised revisions, okay, how you write the essay. It will be useful. This partly related to the presentation part in section D, is that you should have different headers, okay, to lead the paragraph. So to help the examiner to navigate through your essay. So it's important to structure your flow of your essay, okay. So yeah, so as Ms. Rawat has mentioned, you can label it into different layers, investigation 1, 2, and 3. Because for biology EE, because it's quite a large essay, sometimes it's one experiment leading to another. So that's what we mentioned about the layers of your flow. Okay, so layers of the flow. So headings is very important. Okay, and it must be logical as well. Okay, and yeah, and then later on you need to in criteria C, okay, you will look into the evaluation. We will talk about it a little bit more, but you need to mention the source, okay. So it's the source of your data, okay. Is it reliable? And then also measuring equipment, so accuracy, precision. This relates to whether you have chose a suitable apparatus with a suitable uncertainty. So in the previous video I've mentioned, if you have measuring the mass and you know that the change of mass is very small, then when you choose your electronic balance, then you will choose the one with can read up to 3 decimal places. It's much more precise compared to another electronic balance that can only read up to 1 decimal place, because the impact of uncertainty is quite important in terms of your result. Sample size, things like that, okay. And topics, examples, okay. So topic is very broad, and then next part you narrow down to research questions, okay. Yeah, so as you can see in the previous example of the soil bacteria, okay, so looking at distribution of in this case they change another halophyte, okay. So and then C ester, and then they include the binomial name of it, okay, and look at soil salinity. So and that approach, so it would be good to have some planning. So because throughout the EE you need to communicate with your supervisor. So it will be useful to use a table format to show the progress of your EE. Okay, so and so on. Okay, and then aware of double dipping. So the topics that you do in biology IA should not be the same as your biology EE. So they should be completely different. Okay, so avoid double dipping. Okay, and then here I also highlight some of the differences between biology EE and biology IA. Okay, so the EE is more look into the theoretical framework, so how you construct your arguments. So using the reference that you have obtained and your experiment. So that's why it's very lengthy in terms of words. Whereas for biology IA, it's more like a lab report, okay, a more thorough lab report. Yes, so again, here you can see that it's the EE focus on students' ability to analyze and evaluate scientific arguments. Okay, so it's like the theory behind it. Okay, finally we come to the section A. Okay, so so these are the things that you need to watch out for. Area research. Okay, significance, scope of study, focus of the study. Okay, so yeah, another thing to mention is like why your experiment worth to investigate. Okay, so the significance of it. Okay, so and then the other two you've mentioned already. And then when you construct your research question, first of all, you need to make sure that it's in question format. Secondly, you need to mention your IV and your DV, okay, and include some CVs in your research questions. And it will be good to mention how you measure the DV, okay, so more more specific RQ, okay, and then binomial name, a reminder for binomial name, the genus must be capital, species is small letters, and it should be italicized, okay, it should be in italic, okay. So one of the feedback from examiner is that student didn't form a proper format of the binomial name, okay, and then CV, okay, so within time frame. So these are the things that they're looking for. Research question, answerable, so based on your resources, time, okay, and then clearly again in academic writing format, and then you should mention your topics and your research question at the start of your essay, okay. So usually some students, they will use it as a heading, place their research question as the heading of their essay. It's best that you read this on your own as well. Okay, so I'm just focusing on some overall briefings. So here also we mentioned that like when you talk about your methodology, you need to explain how it answers your questions, justify your choice, okay, explain along the way. And yeah, I think that's about it for section A. Okay, so again, you can pause the video, look at the link description below, you can download the PDF, and then read it for yourself. Okay, so next one. Next part I want to talk about is the positive and the negative control. So the negative control is related to how the IV, your independent variable, causes the change, which is the result. So for example, let's say the effect of green tea, okay, on plant growth comparing to no green tea in plant growth. Okay, so that is your IV, right? Present and absence of green tea on plant growth. But the positive control, okay, is to check if the method or apparatus are valid or any flaw. So for example, you need to make sure that, for example, you get it from the same batch of seeds, okay, so the level of freshness, and then and for example, how you measure the plant length, okay, so for example, at certain part of the plants, from the roots to the tip of the plants, okay, so the top leaf in the plants or things like that. So you need to state how you measure it. Okay, so this is more, the positive control is more related to the method of or the apparatus that you're using. Are there any flaws into it? Are they valid? So a very good example is such as the pregnancy test strip. Okay, it's not that it's just an example. It's not that you should be using pregnancy test strip for your EE, but it's just an example to illustrate the idea of positive and negative control. So pregnancy, so this is what happened. The strip is dipped into the urine of the test subject. Okay, and that's why there's a stop line here. And then if it's negative, okay, even for negative result, that means non-pregnant. Okay, you will still see a line on the control line to show that, oh, actually the experiment is working. Okay, so next one, positive test result. Okay, so positive test result, you will see two bands. Okay, one is the band for the control line, and another band is whether there is test positive for pregnancy or test negative for pregnancy. In this case, for the positive test result, you can see that actually the person is pregnant. Okay, now this is the in-phase test result. So if you see, let's say the test strip, pregnancy test strip has no band at all, that means that we cannot be sure this pregnancy test strip is still working. Because if it's still working, then it should have a band at the control line. And then another one, surprisingly, you to also look at whether the band is on the result line, on the control line. So even if you got one band, but the band is not in the correct positions, okay, so there's a result line, but then there's nothing on the control line. This is another one showing that the pregnancy test strip is ineffective, okay, so it's not working. So in that case, then you should use another test strip or another brand to redo your experiment, redo your pregnancy test. So that's how you should read it. The importance of positive control. The control line here is an idea of the positive control to check if your method or your apparatus are working. Okay, so as you can see here, the invalid test is when the control line is not shown. Okay, so that's the idea of positive and negative control. And I find some very good videos related to the positive and negative control, and because of copyrights I couldn't play it here, but I've placed it in the video description below. You can watch those. This is talking about how you set up an experiment, and this one is talking about the negative control. I think most of you know quite well about the negative control, but the positive control you need to focus on a little bit more. So you can watch these two, the first one and the third one is recommended. And the next one. So after we've taught a lot of things, so let's summarize what we've learned. So for the topics, make sure it's biology related, and you need to show that, okay, you also need to mention the context, why you need to do this experiment. Is your research question worthwhile? Okay. Again, from the list, if you pause the video and read closely, they mentioned that the layout, you should have the title. So it helps you to narrow down the scope of your study. And then the research question, again, need to be very specific. As you can see previously, you need to mention the IV, DV, and CV. Okay, and in terms of the DV, best to briefly mention about how you measure the DV. Okay, and then related to the research question score band, your discussion, so the background, the knowledge, okay, and the relevant research should be related to your research questions. Okay. And then next part is the methodology. So the method must be, of course, addressing the RQ as well, so related to RQ. And so that is the method part. You need to elaborate and explain along how it works to answer your questions. And you need to justify your method as well. So why you use this method, or why you use this concentration, and so on. And then second part is related to the theory. Okay, so show how you select and use your reference. Okay, oh sorry, there's a typo there. Reference. Okay, so so for example, you obtain the reference from a reliable resources. Okay, and then you use those reference appropriately. Okay, and then later on in the criteria C related to the critical thinking, you will assess whether these references are reliable or how well they apply to your experiment. So which will be covered later on. So another one is the research, some of the suggested website to start your research. I will also post this in the video description below, the links. Google Scholar, some students, they're not very aware of Google Scholar. So Google Scholar compared to normal Google search is that Google Scholar, they will focus on published data. And a good thing about the Google Scholar is that they also have a function where it shows the MLA citations, so you can just highlight and put it in your essay. But again, make sure you double check it, because sometimes the MLA format is not the most updated one. So but it's a good start. So this is a Google Scholar. And then PubMed, okay, some of the medicine related part, Nature more related to the biology part, ResearchGate, Plant Science. The last two part, okay, is more onto the experiment part. So if you're looking for experiment idea, I suggest you to go for the Plant and Science for School and the Royal Society of Biology Nuffiel's part. This two is a very good one. So stay positive, you got this. Okay, so at all, I hope this video helps you to guide your way through in section A in IBEE. Thank you and bye bye.